Hi everyone, I'm really happy to have the opportunity to be here this afternoon and to introduce you to the GenIDA participatory database. It's an international participatory patient registry that about the manifestation and the natural history of genetic forms of intellectual disability with or without autism and epilepsy. The purpose is to accelerate knowledge about these genetic forms of ID and to improve patient care and families' information. The website is dedicated to people with manifestations of ID, to their families, and also to doctors, researchers, and other health professionals that are involved in the management of such diseases. So it was initiated back in 2016 by Jean-Louis Mandel, who recently received a Kavli Prize for his work in neuroscience. And we are collaborating with a lot of clinicians to design the questionnaire and also to analyze the data that we are collecting. We are collaborating also with a lot of national and international patient and families associations. And we are part of the French Rare Disease Cohort program led by the INSERM, who helped us to comply with all the ethical requirements in Europe. So basically, GenIDA consists in an online questionnaire that is actually available in 8 languages, and further translation are in preparation. And it consists in 46 major questions with sub-questions, including 5 free text questions and 41 MCQs. We are asking about medical information, but also behavior, quality of life, adverse effects of some medication. And the questionnaire is meant to be completed and updated by the relatives of an affected person, or even by the affected person herself if she's able to do so. And of course, the data are anonymized. So some numbers, we have currently more than 1,800 participants that have filled the questionnaire. This represents more than 60 nationalities, and we have more than 200 clinicians that are registered on the registry, and we have access either to the world data, all cohorts included, or that are specifically linked to a cohort to have access to specific data. Here you have some of our major cohorts. The main ones are the Coulen-Vries and the Kleefstra syndromes, because at the very beginning, Titske and David Coulen were involved in the recruitment of participants. But we also have a lot of nice developing cohorts, and some that are still quite small, but developing as well. Some words about the Kleefstra syndrome. So last week when I checked, we had 207 participants that have completely filled the questionnaire. 51 had the deletion, 78 the pathogenic variant in EHMT1, and 38% still didn't specify either the deletion or the variant. And here you have an overview of the main items that reported by the families. Such kind of visualization are available online on our website, and they are updated every 24 hours. And you have visualization for every single question we are asking in the questionnaire. And I won't say much more about the Kleefstra syndrome because you will hear about it and about the GenIDA data later on in the afternoon. So I just wanted to quickly give you an overview of the database. So we are recruiting patients, as I said, via direct contact with national and international patient association. And we have several tools also to help us. We have flyers that have been translated into 8 languages. You have them at the registration desk, so please catch them. And we also have posters for clinicians' waiting rooms and rare disease centers. And we are also recruiting a lot of patients via the social networks and mainly via Facebook. Clinicians that are using the data can submit additional specific questions to subset of patients, cohorts of patients. They can also recruit patients for a research project or clinical studies. And the ultimate goal is to generate a new medically significant knowledge about diseases that can be used to either publish scientific articles and also to feed the drafting of national and international guidelines. And the data collected by GenIDA have already been used in several guidelines, notably in France. So I wanted to show you what can be done with GenIDA on a large cohort. And I will talk about the Coulon-de-Vries syndrome that is caused either by a deletion or by a pathogenic variant in CANCEL1. And by the time we had a closer look to the data was in May last year, and we had 237 participants with almost 83% having the deletion and 40 patients having a pathogenic variant in CANCEL1. And by that time, there were only small mentions of respiratory problems in the literature or in the guidelines. And actually, when we had a closer look into the GenIDA data, we realized that asthma and pneumonia and also other respiratory infection were among the most reported comorbidities for people with KDVS in GenIDA. And these problems are considered as major for many families. And at the very beginning, when we actually launched the questionnaire back in 2016, the question about respiratory problem was not that developed, and we didn't mention actually respiratory infections or pneumonia. But we got those information via the open fields that parents can fill freely. And here you have some examples of such open answers in a variety of language. And now we have to understand the underlying mechanism of such problems that have been reported and to make a recommendation for the prevention or even for the treatment of such comorbidities. We also had a closer look to epilepsy in KDVS, and the data we've collected are in compliance with what was published in terms of frequency of epilepsy and age of onset, but we also provide more detailed information about the types of epilepsy found in individuals with KDVS. We also had a small study about the frequency of use of different anti-epileptics, and by that time we had data from 81 epileptic patients, and what came out is that the two most commonly used anti-epileptic drugs were levetiracetam and valproate, with a trend towards a better efficacy and lower side effects for valproate. And as I said, GenIDA can also be used by clinicians to ask new questions and to go into more detail for several aspects of the disease. And this is what we've done with people in Israel that wanted to have a deeper insight into the ophthalmological manifestation of the KDVS syndrome, and also why not uncover novel ocular manifestations of the syndrome. And we launched a study for 6 months in English and French only, and we got 67 participants that replied. They had to fill in an ocular survey and also to send all the medical records, ophthalmic exams, doctor summaries, doctor visit summaries, test results, clinical photographs. And what came out of this study is that the common findings associated with KDVS include amblyopia, strabismus, refractive errors, and upper eyelid ptosis, And we also discovered possible association of the syndrome with some rare retinal disorders and also with nasolacrimal disorders. So the people then recommended to have a thorough ophthalmic evaluation by the time of the diagnosis and then recurrent testing and evaluation. So I hope I did convince you about the importance to fill in our questionnaire or to register as a clinician on Geneida to get access to our data. You have our website on the screen, and you also have our email address if you want to know more. And with this, I wanted to thank all our partners, institutional and funders, and our collaborators that are involved in the analysis of the data, and of course, all the patients that participate, and the Patient Association and the Rare Disease Networks. And thank you for your attention.
Thank you, Pauline. So, do we have any questions for Geneida?
Thank you for this nice presentation on what we can do with collecting the data. You said that the access for the data will have the clinicians. What about the research institutes or research?
Yes, they can also have access.
So do we need to ask for the permission, or how
you
it
just
is?
have to register and then to send us an email saying which court you are interested in, and we'll open the access.
Thank you very much.
You're welcome.
Any other questions? Dimitris?
So my problem is when working with some rare disorders that there is not so much information available, and some of the labs that are not experts in the gene, sometimes they call some mutations pathogenic, which we know that are not pathogenic. And so people sometimes are misdiagnosed, and then they may enter such registers and etc. And so my question is, how do you check for this, like whether they really have this correct diagnosis, or it's more like of patients provided that they believe they have it?
We have, in fact, we don't have the authorization to ask for the diagnosis. So, yeah, and we have to rely on what people are entering in the database. And sometimes they're coming back saying, okay, I have another diagnosis. So, yeah, we have to rely on it.
Any other questions? Okay. Thank you, Pauline.
You're welcome.